It's the same. Anyway, I guess it's about time that we should get started. It's about 1 after. I want to be mindful of everyone's time. Welcome back to the Building Bridges Across the Laboratory Community series. We're so glad to have you all here today. As a reminder, this series is being launched in collaboration with ASCP and the CDC as part of the One Lab initiative. We're at the midway point of our series now. We really hope that these case studies presented thus far have really sparked your interest on how we as the laboratory community can continue to build bridges to tackle public health emergency responses and laboratory workforce challenges. I'm Allie Brown. I'm the Chief Officer for Medical Quality at ASCP.
And good morning, everybody, or good afternoon, depending on where you're from. I'm Dr. Rodney Rohde. I am at Texas State University, and I'm a medical laboratory science program chair and Regents professor in the College of Health Profession. It's my honor to assist ASCP and CDC with these Building Bridges programs. Today's case studies on the National Wastewater Surveillance System will be presented by Dr. Amy Kirby. She is the lead for this unit with the CDC. The NWSS has been used for decades to identify communities with polio transmission. However, the COVID-19 pandemic, as we all know, presented some new opportunities to modify the surveillance approach to detect COVID-19 infection trends, inform public health messaging, resource allocation, and even personal health recommendations, like masking, for example, or traveling. We would also welcome the opportunity for Dr. Kirby to speak to the types of lab-to-lab and other partnerships that support this incredible resource.
Thanks, Dr. Rohde. Before we get started, we have a few housekeeping notes. All lines will be muted, so add any of your questions along the way in the QA chat box, and Dr. Rohde and I will moderate those questions in the second half of the session. Closed captioning for this webinar is enabled for any interested parties, or interesting, hopefully you're both, and presentation slides, a short summary of the case, and the recorded session will be available in about 2 weeks. Finally, we'd like to acknowledge that this resource was made possible by funding from the CDC. However, ASCP is solely responsible for its content, and the viewpoints we express today do not necessarily represent those of CDC. With that, we'd like to ask Dr. Kirby to go ahead and introduce herself before we get started on her presentation. We're very excited to hear about this collaborative exercise and to hear and learn more about a part of the laboratory that we might not think about very often, but that affects us every single day. Dr. Kirby, over to you.
Great. Thank you, Allie and Rodney and everyone else for inviting me. It's always good to talk about wastewater over lunch for those of us that are on the East Coast. Hopefully you will enjoy this talk. I also, as I was listening to the intros, I noticed that my audio was going in and out. So if you guys are having trouble hearing me, if people can just shoot me a message in the chat. I've got my phone and I'll transfer over super quick. But hopefully we won't have that issue. So what I want to do today is provide a little background on wastewater surveillance. I don't know how many folks on this call have any familiarity with that approach. So give a little bit of the justification behind why we use it. And then talk about how the news program has been implemented. I wish we were decades old. We are not. The news program was actually established in 2020 as part of the COVID response, so much younger, but give a little update of where we are since we've been implemented. And then I want to focus on the addition of MPOCs to the system, which I think really highlights some of the opportunities to work among different types of laboratories. And also talk about some of the innovation that we're working on and supporting as we move forward, because that has a lot of collaboration across different types of laboratories as well. So this is my usual intro slide. This is what I always want to use as my intro slide. And I'm going to turn off my video so we can get better bandwidth. I feel like this is how the majority of the public health world feels about wastewater surveillance at this moment. And it's either you fall on the side of this is a great new tool and we should be using it as broadly as we can, and we're so happy that it's so hot right now, or it's a very dismissive, like, oh, wastewater surveillance, it's just the hot thing right now. And
Oh, Dr. Kirby, I don't think you shared your slides. We can't see the slides.
Oh
right now.
no, that is an important step. There we go. Better?
Yes, thank you.
Okay, sorry about that.
We can't miss the Zoolander reference. We have to have it up there.
Yeah, you can't miss that. So this was my real title slide. This is the one I would prefer to use. Yeah, so as I was saying, I think everybody feels this way, and sometimes it's good. This is an exciting thing for this new tool, and sometimes it's just so much talk about this, and it's not really proving to make a big difference. I hope in the next 20 minutes I can convince you that it's more on the former than the latter. But either way, a little Zoolander reference never hurts. And it has, in addition to the talk here in the US, it's really taken off globally. We have been doing wastewater surveillance for polio for decades, but it really hasn't been used outside of that framework until COVID. And then with COVID, it really took off. This is a dashboard of dashboards called the COVID Poops dashboard, which came to be in 2020. When it first started, there were less than 10 dots on this map. And you can see that it has really grown significantly in the past 3 years. So lots of uptake of this type of tool. As has already been mentioned, this really started with polio, and I like to talk about the polio example because it's a really great example of why wastewater surveillance is important and why it offers a different approach than other types of clinical surveillance. And so this really came to be as the polio vaccine was being rolled out, and the initial thoughts about potentially eradicating polio were really first coming to be. And if you're thinking about eradicating a disease, you really need to be able to track down every single last case to be able to contain it. So what kinds of surveillance do you need to do to be able to track that case? And they knew that polio presents a really difficult problem for clinical surveillance. So if you have 500 individuals that are infected with the polio virus, only one of them, or not even one, depending on the type of virus that you're talking about, will actually have acute flaccid paralysis. So what we think of when we think of polio case, so the most severe manifestation. Another roughly 125 people will have some symptoms, but they tend to be fairly mild. Some of them will have some neurological symptoms that don't get to the point of paralysis. Those will probably get diagnosed. A lot of them just have gastrointestinal upset. Like everything else GI upset, that is very unlikely to be, they don't even go to the doctor, much less diagnosed with the specific virus causing it. And then the rest of those 500 people that are infected, they're asymptomatic completely. And so they're going to be completely silent and invisible to clinical surveillance. But what are all 500 of those people doing? They are shedding poliovirus in their stool. And so by looking at wastewater surveillance, we can get information from all 500 of these people, as opposed to just that subset that are having symptoms, and even more likely, the subset that are having very severe symptoms. So when case ascertainment is very low, as it is for polio, wastewater surveillance has the potential to have a lot of applicability to help us drive interventions. And with polio, we've seen that that was extremely successful. So polio wastewater surveillance started in the 60s. A formal eradication initiative started in 1988. 2 wild-type polio viruses have already been eradicated. We only have 2 countries with endemic wild-type polio virus as of last year. As we know, the vaccine itself has caused some issues, and there are some vaccine-derived polio virus outbreaks. But overall, this has been a very effective way to identify those neighborhoods where polio virus was circulating, and we could target vaccine campaigns to those areas. And that's pretty much where the science of wastewater surveillance stood up until 2023 with SARS-CoV-2 and that emergence. And the reason we initially thought very early on about wastewater surveillance was because of what happened in the SARS-1 epidemic. So when that epidemic started, there was a very well-known case of transmission within the Amoy Gardens apartment complex, which is shown here. This is in Hong Kong. And it was extremely well studied, and really the only viable way that this transmission could have occurred is through the wastewater system in this apartment complex. So the apartments are connected by their wastewater system vertically. So you're connected to the apartments above and below you. And there was a case that got infected with SARS-1 that had not left their apartment for months, did not use the elevator, did not go out in the hall, homebound. And so it was very puzzling how they could have become infected, but it was shown quite clearly in this apartment complex that there was a connection from a case on a lower floor, and that likely aerosols generated through the wastewater system had traveled up to that homebound individual and caused the infection. So transmission through wastewater, while we think this is very limited, is possible. And so when SARS-CoV-2 happened, there were a handful of folks that remembered this case quite well and started wondering, one, about transmission through wastewater, which luckily with SARS-CoV-2, we don't have any evidence that that happened. So we think that is the likelihood of that is very, very low. But secondarily, is there a potential for wastewater surveillance? And with a lot of work across a variety of laboratories, academic labs, public health labs, commercial labs, we were able to show that we collectively, the field was able to demonstrate that if you measure SARS-CoV-2 RNA in wastewater, those concentrations do track with trends in cases, hospitalizations, and severe outcomes. So it is giving us an indication of what's going on in the community. And even more importantly, we're able to detect those trends in wastewater earlier than we see them in case data or hospitalization data. So wastewater trends on average lead trends in cases by 4 to 6 days, which in a fast-moving epidemic like SARS-CoV-2, that is a lot of time to do something earlier. So this is a very basic schematic of what wastewater surveillance as we do it for SARS-CoV-2, how that works. So individuals use a toilet system on a sewer. We can talk about applications outside of sewered systems, but our focus here in the US is on these sewered systems. They flush the toilet within about 3 hours on average. That wastewater arrives at the centralized treatment plant. Operators there collect a sample. Ideally, it's a 24-hour composite sample, so we're representing everything that flows into the treatment plant that day. But we can also use grab samples, which are just like a single point in time for that day. So they're just a bit noisier data-wise. So they collect that sample. We were initially collecting about 1 liter. Now we're down to collecting 100 mils per sample. So we've gotten that sample size down, which helps considerably. That is then sent to a laboratory where the sample is processed, so concentrating the viral particles out of the wastewater, extracting the RNA, and then quantifying the RNA using either real-time or digital PCR. That data is then submitted as raw data to CDC. Our decipher system, which I'll show you in just a minute, can analyze that data and provide information on trends that can be used. Those metrics can then be used to inform public health decisions. So what are our health departments doing with this information? What we hear most frequently is that they value it as an independent confirmation of increases or decreases in cases in a community. So what is really going on in the community that's not impacted by all the noise that impacts clinical surveillance data. So it doesn't matter if people are getting tested, it doesn't matter if people are using home tests that aren't getting reported anymore. It does not matter if clinical capacity has reached its peak, which we saw happen unfortunately all too frequently in 2020 and 2021. Wastewater data will continue to report on that overall community burden estimate, totally independent of what's going on with clinical care. They're using this information to inform public-facing dashboards. We have anecdotal information that individuals are using this information to make decisions about their healthcare. Are we going to mass today? Am I going to go to that large event? Should I travel? We are, of course, informing public health leadership. Early in the response, they were using it to make decisions about siting of testing capacity and vaccination clinics. Now what we see them using it for more is that near-term forecasting of cases and hospital utilization. So can you identify communities where increases are happening and reach out proactively to their hospitals and make sure that they're prepared and have all the resources they need when some of those cases show up in their ER in a week or two. And we can also use it to detect the emergence and spread of variants of concern. So that's a super quick overview of sort of the science of wastewater surveillance. As I said, we implemented the National Wastewater Surveillance System, or NEWS, in 2020, and it's grown very quickly since then. So on the left is a map of all of the jurisdictions that are using CDC funds to support wastewater surveillance. As of May of this year, we have 48 states, 5 cities, and 2 territories that are funded to participate in NEWS. We've also established 4 centers of excellence, indicated by the stars here, and those are based at a health department and have, as part of their required consortium, an academic partner, which generally is a lab. And then also a utility partner, because our utility colleagues are really important for understanding the data and making sure that our sampling is as robust as it can be. That state-led system is then supplemented by a commercial testing contract, so a commercial laboratory partner that CDC manages, and that provides an additional 500 sites nationwide to give us capacity to rapidly add testing as states are setting up their systems, to be able to surge in the event of some kind of response that we need this for, as well as provide direct support to tribal communities, which we can't do through our ELC mechanism, funding mechanism on the left. Collectively, those 2 systems represent over 1,400 sites. And in those 1,400 sites, we can get data on a population of over 144 million people. So over 40% of the US population already. And I can see I'm way behind, so I'm going to speed up. So this is the Decipher dashboard, and it's really the heart of the system. So as I said, we measure the concentration of RNA, but what we're really looking at in wastewater is these higher order metrics, because the concentration that you measure in a sample is influenced by the design and operation of the wastewater treatment plant. And so you can't compare concentrations alone directly across jurisdictions. So we use these metrics to be able to enable those comparisons. The percentile metric is where are we now? So looking at the current concentration, how does it compare to all of the concentrations measured at that site? So is it among the highest 20% ever detected, the lowest 20%, or somewhere in between? So where are we now? And then percent change is where are we going? So for COVID, we're looking back at the past 2 weeks of data and asking how quickly are these concentrations changing, and are they going up or down? So between the two, you can see, are we high or are we low, and are we going up or down, and how quickly is that changing? We have expanded the system. Shown on the left is our original COVID data tracker page. And this is our percentiles and percent change metric that I just showed you for SARS-CoV-2. In the summer of 2022, we added the ability to track variants, and there's a COVID data tracker page specifically for that. And so you can see what the predominant variant is and how that changes over time. The distribution of variants changes over time. And then most recently in October, we added mpox testing. I'm going to talk more about that in just a minute. So I will not go into a lot of detail there. I also want to quickly note that we sponsored an objective review of wastewater surveillance for public health through the National Academies of Sciences, Engineering, and Medicine. And this is their summary of their phase 1 report. And I really want to focus on that second bullet, which is they define a robust and valuable national wastewater surveillance to have these characteristics. It should be equitable, meaning that the use of this tool should not be dictated by the demographics of the community. So it should be available to all communities, even those small and rural ones where it might be harder to implement. It should be sustainable. This isn't a system that we want to turn on and off for responses. It is most effective when it is running consistently. It needs to be integrated, which I think is really important for this conversation that we're having today. And by that, they meant integrated vertically, so federal, state, local, as well as horizontally, so between jurisdictions and across different types of partners. Needs to be actionable. This is not data for data's sake, but data for action. So how are we going to collect information that can inform public health actions? And it should be flexible. This is not a surveillance approach for a single disease, that we are no longer talking about just polio surveillance or just COVID surveillance. This is a surveillance platform that can be used to address a lot of different public health issues. And so it needs to be flexible enough to respond to changing public health needs. And the report on that is available online. If you're interested, you can look up NASEM and wastewater, and it should take you right to it. So how did we implement this for mpox? And I wanted to highlight this activity because I think it really gives a good example of how different types of labs can contribute and have a really critical role in supporting the flexible integrated wastewater surveillance system that NASEM outlines. So mpox was truly implementing the unexpected. So when we first started seeing cases in May of last year, it was not something that we had considered as part of news. We were thinking about a lot of other pathogens. Mpox was not on our list at all. But we very quickly started getting questions about whether wastewater surveillance could be useful here. And to be quite blunt, we had no idea. There were no studies looking at shedding of mpox virus in feces or urine. We had some idea that the dermal shedding from lesions could contribute through like showers. But what does soap do to that? Like, we just had no clue whether this would work. And so we really had to reach out to all of our partners and learn what they were doing and try and get all of the information that we could. And there is a consortium of academic, commercial, and philanthropic partners called Wastewater Scan that runs out of California. And they, because they have an academic partner, they can adjust really quickly to implement some straight research tests, right? Just see if it works. And so they were providing wastewater surveillance data for COVID for many of the communities in California that were having mpox cases. And so they were able to take samples that they had gotten in for COVID testing and start trying to see if they could also detect mpox. And so they were able to do that very quickly. And within just a few weeks, they had evidence that, yes, we can detect it. Yes, we are in fact detecting mpox and not something else in this very messy matrix of wastewater. And that the detections for mpox correlate with cases, which is all great news. But this was a research-based test. So that was enough information to say, yes, this is worth pursuing, but it was not enough information for us to just take their assay and run with it. So then we had to turn to our commercial contractor, which is the BioBot company, and work with them to validate a test for mpox. There was some changes in the type of assay that we wanted to do. So instead of the mpox species test, we wanted to move up and do the orthopox test because of changes in the way the select agents program viewed those tests, get that validated. And then once we did that, we could get it implemented at the 500 sites that are participating in our commercial testing contract, which is wonderful. And so now Wastewater Scan has also implemented this in their sites. So we get data on over 500 sites twice a week. We've got over 24,000 samples tested to date. I'm very happy to say that only less than 3% of those samples are positive. And in the last few weeks, we've started getting weeks that go by with absolutely no detections, which is wonderful. We're also working very carefully with the response to do wastewater surveillance in areas where we know we're seeing some more cases. So this has proven to be really helpful to confirm that we aren't missing clusters in significant cases somewhere that clinical surveillance is not seeing. I'm going to skip over this for time because I want to focus on this continued innovation in wastewater surveillance. So this is although it's an older field for polio, this type of quantitative wastewater surveillance at scale in sewer systems is very young. And so there's lots of innovation, and at CDC, we really want to engage with that. And so I wanted to share some examples that really highlight that innovation. So one is up there on the top left, these on-site testing systems. This is really partnering with our clinical laboratories and clinical partners to look at the assays that they're developing, particularly around the COVID response, and do they work for wastewater? Some of you might be familiar with this kit. This is the Cephia Gene Expert Kit. It's an all-in-one cartridge. So you just put your sample in some sample buffer and add it to the top of that cartridge, flip the cartridge into the machine, and it gives you a quantitative result in about an hour. Amazingly, it works just as well for raw wastewater. So you can just directly add raw wastewater and have a sample result in an hour. And it works quite well. It is not as sensitive as a full assay, but it's really important to have this type of capacity for places that are very remote. For example, we're implementing this in Alaska, where they would have to charter a plane to get a sample to the nearest laboratory. So this is a way for them to participate without having to bear the cost of chartering multiple planes a week. We are looking at flexible sampling options. So if you can't do those compositive samples at a treatment plant and you want to be able to sample out of a manhole, how can you do that? There are passive samplers that a lot of our academic partners have been working with. So continuing to follow that research and develop guidelines around how you can deploy these passive samplers to sample out of manholes where other types of sampling might not be feasible. We have used the Small Business Innovation Research Award program to drive innovation in the biotech sector. So we really want a preservative for wastewater that allows us to ship these samples at room temperature without losing signal. And we're really thrilled to see that Zymo has been able to get early data suggesting that they do have a product that will work for this. So that will significantly reduce shipping costs to be able to do this at room temperature instead of on wet ice. We're working with NIH, so they have a program, the what's it called? RADx Tech program that drove innovation around COVID diagnostics. One of the things that came out of that was the series nanoparticle concentration method. And it turns out series nanoparticles work really well for pulling viruses and other pathogens out of wastewater. And it allows you to do wastewater processing at scale, which you couldn't otherwise do. So working directly with series, working with NIH, working with the academic and public health labs that have implemented these series systems so that we can understand how they work and really work them into our workflows. We're also working on improved data submission. So ideally, we have an API implemented within Decipher. And so we want to have direct links to state labs so that when they're doing this testing, no one has to sit at a computer and do manual data upload. With direct data links, that will just happen automatically and reduce the reporting burden while also shortening the reporting time. Real quick, where we are going next is we are expanding this to really take advantage of the fact that this is a multi-pathogen surveillance system. So right now we are validating our news panel version 1.0 to expand to non-response targets. And this is all of the pathogens that are being validated for this panel. So we're adding normalization and process controls. These are already part of our required elements for data submission, but those will, of course, be validated within the panel. So pepper mild mottle virus and crassphage are markers of human fecal contamination that we use to normalize the data. And then bovine coronavirus is a process control to get recovery efficiency. We're adding a panel of antibiotic resistance genes with targets based on the AR threats as identified by CDC and WHO. And then the pathogen targets have expanded considerably. So the respiratory panel, in addition to SARS-CoV-2, will have influenza and respiratory syncytial virus. We have a limited enterics panel, and I'm happy to talk about why that is limited right now. Adenovirus 4041, Shiga toxin-producing E. coli, Campylobacter and norovirus are on that panel. And then an emerging pathogens panel, which will continue to have Mpox on it, but we'll also add the fungal pathogen Candida auris. And this is also a really good example of collaboration across laboratories. So we have a contract with a commercial laboratory, GT Molecular. They are currently optimizing these assays. And so what that means is they are multiplexing them to the greatest degree possible so that we can make this as cost efficient as they can be. And doing that for both of the common digital PCR platforms. Once we have those optimized assays, it will come to CDC for validation internally, and we will use that to develop open source protocols. We will then implement those validated assays within our 4 news centers of excellence. So they'll be the first to test out this panel. And then assuming all goes well, we rolled out to the full system within 6 months of that. So looking at probably summer of next year. I do want to note this is not a single point in time expansion because we want this to be an adaptable and flexible system. We see this as an iterative process. So every year, the news team and SMEs, subject matter experts from across the agency will review the panel components. How well are they performing? Are these the public health needs that we need to be focusing on? Have things changed? Are there other targets that we need to consider for inclusion? So this will change routinely as public health needs change so that we can make this platform as impactful as it can possibly be while also doing it in a sustainable way within our public health network, particularly the laboratories that will be managing all of this testing. And with that, happy to answer questions.
Thank you so much for that tremendous presentation. I mean, there's so much going on as usual that we, when you look at other parts of the laboratory community, it's amazing to see how much work really is going on behind, and this, no pun intended with the behind there, but yeah, excellent work. Excellent work. We do already have a question in the Q&A, and I'll remind all of our participants that you can enter any questions you might have in the Q&A section of Zoom. The question is, since transmission of viruses needs to be monitored from one country to another through travelers, do you think checking the lavatory wastewater in airplanes should be included in national surveillance? It's a great question.
Yeah, so CDC does have a travelers genomic surveillance program that includes lavatory monitoring in planes. The challenge with that is human behavior, actually. So we will only get data where people poop, to be very simple about it. And so you have to think about how people's bathroom behaviors are influenced by where they are. So homes are always preferable to workplaces. Schools are a little bit, kids don't seem to have the same kind of inhibitions that adults do. So schools can be a little better about that. But planes are an example where it's like most people are going to avoid going to the bathroom on the plane. And so you're really only capturing quite a small subset of the people on the plane. So you always have to keep that in mind. However, it has been shown to be quite useful to look for potential new variants coming into the US. So the movement, the importation of variants, it's combined with pooled traveler screening. So off of a plane, you look at both the lavatory and you take pooled nasal specimens from all of the passengers that are willing to be tested. And then combining those two data sources together, we feel like we actually get a really good view of importation of new variants. So, and we're talking about now, could that be expanded for other pathogens as well? So more to come on that.
It's an interesting question, and it makes me kind of think as a follow-up, perhaps the airports themselves for major, hubs, maybe looking, because people might get off the airplane and actually be more comfortable with using the facilities in a building, so to speak. But just that's an interesting question. I hadn't really thought about that. Could you talk a little bit about, Dr. Kirby, about some of the partnerships that you brought them up through your talk, and I'm always interested. My ears perked up when you talked about Cepheid and kind of the clinical side of that, and I've also got a background in public health. So those partnerships, as you know, are so critical. Can you talk about that with respect to really anything, whether it was COVID or Mpox or otherwise, that you really thought was impactful? And did you run into some hurdles and disadvantages around that when you were dealing with partners outside of, say, the federal government or state health departments?
Yeah, I mean, the lesson that I have learned and that I try to keep top of mind as we continue to develop this is that this is going to sound weird to this group. Not every laboratory is the same, right? We all have different capacities and different strengths. And so what I always try to make really clear to the team, to the news team, is we need to keep up with what everyone is doing and sort of what their motivations are and what their strengths are, because you never know where the next innovation is going to come from. And so Mpox is a great example. When we started thinking about trying to validate and do some of this testing in-house for Mpox, CDC does not turn on a dime, even in the space of a response. We had significant delays to be able to do Mpox testing in our news lab. And so we couldn't even validate the test on basic samples before wastewater scan had already basically solved the problem. And so we needed to be cognizant of that. And so now that's sort of our approach. We don't even start with trying to do it internally because we know that external labs are going to be able to move much more quickly. I think the other thing that I always like to keep in mind is what are their limitations, right? Not just what are their strengths, but what are their limitations. So when you're thinking about these partnerships, for example, we know when we're reaching out to academic labs and saying, what are you researching now? Can we ask you questions? Can we use this data? They need to be able to publish. And so we need to be able to address that up front. We are not going to publish your data. We're not taking it. It is yours. We're going to protect that so that we're all on the same page. Same thing with Commercial partners, we need to be really clear. We are not endorsing your method, right? We want to evaluate it from the ground up. If it's great, excellent. If it's not, we're not going to move forward with this. And so we need to recognize that they need to see a market. That's what they're looking for. Public health is looking for what is sustainable within the scale of what we're already doing. So these aren't laboratories that are just going to shut down all of their activities and only do wastewater. They're juggling a ton of other needs, things we don't even necessarily think about. They may be conducting regulatory water testing. They may be doing all kinds of chemical testing for EPA needs. And so being able to balance all of that and make sure that when we do come to them with an ask, it is as streamlined as it can possibly be to work within their limitations. So I think having a good working knowledge of kind of how all of these groups work really helps you to engage with them effectively and know who to go to when you have that question.
Yeah, that's a great answer. We have another question. You kind of alluded to this a little bit in your response from our audience. What do you see for the future of the NWSS? Do you see any possibility for monitoring chemical formulations to identify communities in need of special services, such as drugs of abuse?
Yeah, so we are thinking carefully about particularly illicit substances, although I think chemical testing is another one. So kind of goes back to that previous question of knowing what a laboratory can and can't do. So we're trying to figure out within a public health lab that would be doing this testing, how feasible is chemical testing for wastewater? Is this something they can easily take on or not? So that's a kind of implementation question. The bigger question is, how does wastewater data, particularly for illicit drugs, what are the actions that come from that? Is it interpretable for the kinds of interventions and public health actions that we want to take around opioids? Because remember, you're not getting case data, you're getting overall rates in the community. So how do you understand that? One of my colleagues in overdose prevention here said, what you really need to know is intent of use. And we can't even begin to get at that with wastewater testing. So does knowing overall trends in use, knowing it's a mixed bag, can you interpret that? So we are going to be doing some pilot studies looking at that. The other piece that's really challenging for particularly illicit drugs is the ethical piece. So environmental testing for public health doesn't fall under the same ethical guidelines as clinical testing, like IRBs and all of that. But we still need to be thinking about those things. And whenever you've got something that could potentially be linked to law enforcement, you need to be very, very careful and have some guardrails around that data. So we want to have all of those conversations and establish those guardrails and a clear public health use before we go down that path at scale.
Yeah, I really appreciate that answer with respect to intent and law enforcement. When I was working in public health, I was through the anthrax events right after 9/11. There was always, concern about what are you going to do with that information, because you certainly could potentially place blame somewhere where it's not, right? So you really want to be careful with that. Here's a question that is kind of near and dear to my heart with respect to public health and One Health driven initiatives. So I used to work in zoonotic control within the laboratory as an epidemiologist. Has your program looked at perhaps possible surveillance of animal reservoirs? So I'm thinking, pig farms, fowl, other types of farms that could potentially be sentinels to human disease. We've seen this in the past. We've used in Texas, for instance, chicken sentinel flocks to look at serology for different mosquito-borne viruses and things like that. Have you thought about that within animal farms or places like that?
We have. So animals come up in two different ways. So one is what you were mentioning. So can wastewater surveillance be used to improve surveillance for zoonotic diseases or completely animal diseases? The other way it comes up is, are animal infections influencing the wastewater data that we're getting? So are you picking up human and animal cases? So I'm going to address the second one first. For SARS-CoV-2, while we know there are animals that can be infected, we have not seen evidence that it is skewing the wastewater data in any significant way. And it could be a combination of things. One, the animals that tend to get infected don't contribute to wastewater in large amounts. So what we tend to see in wastewater are things like rats and wild birds, not so much deer, cats, things like that. And so that's mink. We don't see mink in wastewater. So that's, we got very lucky with SARS-CoV-2 that we're not seeing that. But with some of the other targets that we're looking at, there absolutely will be animal contribution. So if you think about something like West Nile has been proposed, with such a huge wild bird contribution into wastewater, we are absolutely going to be picking up wild bird contribution for West Nile. And how do we separate that from human contribution? So that could still be very valuable data, but you have to include it in your interpretation of the data. So we're still sort of working through that, but always a question when you're thinking about a new target for wastewater. Looping back to your initial question, which is how can we apply this to animal pathogens or zoonotic pathogens? That is something that we're interested in doing. Generally, the kinds of animal facilities that we're most concerned about, so large agricultural production facilities, that wastewater is not flowing into wastewater systems. It's being contained on the farm and dealt with that way. There is some discussion of, well, could you test the wastewater on farms? And in theory, yes, you can, but implementation of that, is really up to the farmer. You're asking to be on their private land doing testing. And so I think trying to figure out what the right scale and use of that kind of on-farm testing would be has been the challenge. But it's definitely something that we've been thinking about and how that could be applied.
Wow. Thank you so much. You alluded to at some point in your presentation about equity of testing and making sure that there's access to this type of testing, even in maybe more rural areas, et cetera. I know it's very highly sensitive testing, but are there any considerations for areas that are less populated in our country and the appropriate monitoring of wastewater surveillance?
Yeah, so I mean, it's always voluntary, right? So the community itself has to, maybe not the whole community, most people aren't asking the community, but the wastewater plant operator, usually the city or county manager will be part of the decision of whether or not they're going to take on wastewater surveillance. And so it's not something that we can require of them. The question for us is really about data sharing. So if we have a very small community, who gets access to that data? So certainly the community itself does, we share everything with them. For communities under 3,000 people, we don't make that data public. So it doesn't go up on COVID data tracker because it's such a small community. You could, in theory, have some privacy issues there. We also don't include that in any of the data sets that we share with individuals. So like academics will ask for our data to do analyses. We remove all data for samples under 3,000 people. So those communities will be screened out. If they want access to those, they have to go directly to that community and ask for that data. So that's how we're handling it right now with SARS-CoV-2. It's something that we readdress with every new pathogen because it changes. Mpox had some very different privacy concerns than COVID did because we saw this transmission largely among men who have sex with men. Some of the sampling was based on where we knew there were very dense communities of men who have sex with men. And so there became questions about, could wastewater data be used to stigmatize these communities even more than they may already be? And so there were lots of discussions about that as well. Ultimately, we decided that it was more valuable to make that data public so that individuals could know, with again, that same screen of communities under 3,000, the data is not public.
That's great. That's great. I think maybe possibly one more question as we kind of move towards the end here. I'm going to switch back a little bit to gene expert testing that you kind of mentioned. So again, if you're thinking globally around that, non-invasive sample types like that could obviously be an invaluable option for sort of a point of care test in the field. What do what potential do you think wastewater or personal stool collection might have for those kinds of applications? And I'll also slip in just real quickly. I love your list of the things you're looking at. Have you looked at, or I mean, I'm sure there's a group of people thinking about these, but Hep A and Hep E and Vibrio and other kind of waterborne based things as you kind of think about that?
Yeah. So I love the question about can we do personal stool testing? One of the challenges that anyone who has ever done any stool-based health testing is the stigma around stool is immense. And so getting people to agree to stool testing, collect a stool sample, test their own stool, tell you if they pooped on the plane, like it is really hard to do. So I think the idea of personal stool testing, probably not, because I just don't think there's a market for it. There's lots of discussion of it being a better diagnostic in the clinic. That's probably going to happen. But personal stool testing, just don't think people would do it. However, I have heard some like real pie in the sky discussion about like a toilet that tests your health. That may have a market. I mean, it's going to be very expensive, but like, what if when you went to flush your toilet, it changed color if you needed to go to the doctor? That is technically not a really far off idea. It's very similar to sort of the real-time monitors that people are talking about putting into wastewater lines. It's essentially a real-time monitor in your personal wastewater line. Could there be a market for that? Yeah, I think there could be down the road. It's probably only going to be for the, really well off. But I think it's something that I would not be surprised to see coming on the market in the next 10 years or so. And then the question about, waterborne, let me real quick. These are definitely things that we want to get at. We have not included any of the ones that you named, hepatitis, Vibrios, those in our short list because there are technical challenges. The test that we have available to us right now is too broad to be interpretable. So we're working out the technical challenges, but expect to implement those in the future. Sorry.
It's interesting. No, that's okay. We got a couple minutes. It's interesting, and Dr. Brown can jump in here too. But just one quick thought when you talk about the toilets and the water turning color and stuff, one thing I think we have seen in my lifetime in the last decade that has become more acceptable to the public is, I don't want to do a colonoscopy every day, but I can send in my poo for at least a representative kind of screening of your DNA that's being shed for different markers. So, who knows, the country or the world might be more inclined to do that instead of going to the doctor. So many people just will not go to the doctor. So maybe there is a market down the road. It's exciting, kind of interesting thoughts to think about that.
Yeah, absolutely. I agree.
The AI commode coming to a home furnishing store near you. All right. Well, thank you so much, Dr. Kirby. This has been really, really interesting. It's just so fascinating to hear about something that there's so much activity going on behind the scenes, much like many of us do within our own laboratories, and then this being another aspect of that. So thank you so much for sharing your experience and spending the time with us today. We greatly appreciate it. Keep up the great work.
Thank you. And thanks for the invitation.
I have to resist so many puns that I currently want to say, but I'm not going to. Maybe later after the presentation. So our next session is coming up in 1 month. So make sure to register for it soon, either through the QR code, which we have displayed right here, or on our website. So I'll give you a second to look at the QR code here and to be able to use it on your smartphone or other device. And this next session will feature a case study from Dr. Suzanne Dale on how her team at LabCorp worked closely with CDC and FDA to validate and scale high throughput testing platforms to respond to the emergence of Mpox during the summer of 2022. And this will be our first featured case study from the perspective of a reference lab And it will speak to how these labs are uniquely suited to support rapid increases in testing capacity to support public health responses. So part of this series, we're just looking at all sorts of different aspects of how our entire laboratory community has been and can be involved.
Yeah, thank you, Dr. Brown. And as we transition to the next slide, it really brings to mind, I just left last week the APHL Public Health Laboratory Training Conference. And, I want to thank Dr. Kirby and her team and really take the opportunity to remind everyone that public health and the medical laboratory, the clinical side, are partners. We are a team for global public health. And that's, I think, a message that we all want to continue to share with our audience and with the public. That's really critical, obviously, in these times to do that. So we want to encourage, again, all of our attendees to continue engaging with laboratory peers and staying up to date with cutting edge technology around laboratory trainings through joining the One Lab network. And as we kind of go through these slides here, I know we're jumping through them, and signing up for some of these opportunities for access to the CDC One Lab Reach, which has a QR code link, as well as CDC's new LMS for laboratory training materials. Again, last week I had the wonderful opportunity to sit through a couple of presentations about these two opportunities, and they're just really new and innovative and exciting. You can stay up to date with ASCP's work supporting the One Lab initiative and several new resources in our pipeline, including past and future Building Bridges resources and registration details on our website. So I believe we have come to the end of our session today. I want to thank each and every one of you who have joined us, and please do share this information with your colleagues, not only in your own labs, but on different floors, in different buildings, and across the country and within your own communities, because we really want to get this information pushed out so that everybody can take advantage of it. So thank you all. Have a great day and a great 4th of July weekend.